Hi, this is Gene Williams from Promis Neuroscience. Thank you very much for your interest in Promis. I'll be making some forward-looking statements. Let me start with an overview. Promis is developing a best-in-class portfolio of disease-modifying therapies that selectively target the root cause of Alzheimer's. Specifically, we're creating monoclonal antibodies that target the toxic oligomer variants of beta-amyloid tau and TDP-43. Those toxic oligomer variants are the root cause of disease. In addition, another aspect of building best-in-class therapy is we're applying precision medicine or personalized medicine. There are different strains of toxic oligomers, so companion diagnostics will be critical to identify which strain a patient has and give the right patient the right drug. We're publicly traded on the Toronto Stock Exchange. Our ticker symbol is PMN.TO. Our strategy is built on a very strong foundation. Let me walk you through what that looks like. First is the foundation of the Alzheimer's market. Alzheimer's has such significant unmet need and great opportunity that we've seen continued investment despite many failures, and that's left us all in a situation of tremendous scientific information that can guide our strategies for product development going forward. One of the most important insights that we and others in industry have seen from the work done so far is that the toxic oligomer of beta-amyloid or tau is the neuron killer and the disease driver. And since there are different strains, there's a need for personalized medicine. Well, the second aspect of the foundation is one of the most important strategic shifts in industry that I've seen in my 35 years. When I first got into industry, the key to successful product development was to be first in class. First entrants got the highest market share. Now the biggest products in industry, the ones that have the biggest peak sales ever, are not first in class, but best in class. Our scientific understanding and capability has developed so much over the last couple of decades that now the biggest drugs in the world come from the following playbook. Understand predecessor compounds in the clinic or on the market. Understand scientifically where they work and where they're good and where they can be improved. Then apply tools of rational drug design to create products that are scientifically differentiated. Those become best-in-class products. Three biggest products in the history of the industry followed that playbook in progressively more and more scientifically driven ways. Pfizer's Lipitor was $12 billion. It was the fifth statin 10 years after the first. Humira was the third TNF-alpha antagonist in RA 5 years after the first. Sovaldi, $25 billion and growing in hepatitis C. We certainly believe, and I think we're not alone in this, that best-in-class therapy in Alzheimer's will set new records for peak sales. Now, the other aspect of the best-in-class playbook in industry is none of these products were developed by the very large successful global players who commercialized them. David beats Goliath in innovation. Humira was developed by CAT, then sold to Cambridge, then sold to AbbVie. Sovaldi was developed by Pharmasset and then acquired by Gilead. Promis has a slingshot. David needs a slingshot to beat Goliath. Our slingshot is our technology platform, a rational drug design approach for monoclonals that allows us to identify what we call disease-specific epitopes. An epitope is the target. If you have a target that's only present on a toxic oligomer, you can then design monoclonal antibodies that selectively go after only that toxic oligomer, and you can design companion diagnostics that allow you to apply a personalized medicine approach. This is the foundation that we've used to build our best-in-class portfolio of monoclonal antibody therapies that are targeted to the oligomer, have a companion diagnostic to account for different oligomer strains, and we believe have a good chance to be best-in-class disease-modifying therapies. Let me take you through some of the elements of this in just a little bit more detail. This is a picture of the biology of Alzheimer's disease. APP, the amyloid precursor protein, breaks down and creates beta-amyloid monomer. There are three forms of beta-amyloid in the brain. Starts as monomer, aggregates into oligomers, and then can further aggregate into plaque. Monomers constantly being created when amyloid precursor protein breaks down. For the longest time, the scientific community and industry felt that plaque was the problem, and that misunderstanding led to a lot of the disappointments in early clinical trials. What an overwhelming body of evidence now tells us is that it's actually the oligomer, with the red arrow pointing to it, that is the problem. Oligomers kill neurons and drive disease. This is just an example of some of the growing body of scientific evidence showing that you can kill neurons with oligomers, as that picture at the bottom shows. You can't do that with monomer, you can't do that with plaque. Oligomers or prions are the root cause of Alzheimer's disease. Now, scientific insight like that is most compelling when it can explain clinical trial results, and that's exactly the case. Let's look at 3 major clinical trial results that came out over the last several months. Aducanumab from Biogen, solanezumab from Lilly, and Merck's BACE inhibitor. In Q4 of last year at the CTAD meeting, Biogen reported 2-year results on aducanumab. It's a good product that looks like it'll work, a great step forward. It works because it doesn't bind monomer, but it does bind oligomers. However, because it binds plaque, it creates a dose-limiting side effect, area E, or brain swelling, that not only is a problematic side effect, but it also limits the efficacy of aducanumab because it limits the dose. Lilly's solanezumab failed in the 3rd pivotal trial. It almost had efficacy that was approvable, but not quite. The reason is it binds monomer. There's so much monomer, and monomer is not the neuron killer, that depleting monomer, whether it's with a monoclonal antibody or, as we'll talk about in a second, with the BACE inhibitor, is just too high a hurdle to climb in order to have efficacy. That was the 3rd failure for Lilly's solanezumab. The first BACE inhibitor pivotal trial readout was Merck's product. In Q1, it failed on an interim analysis for futility. Again, the hurdle is so high, there's so much monomer in the brain, that even if you deplete 80 or 90% of it, there's still enough raw material to keep creating those toxic oligomers and driving disease process forward. So step 1 in a best-in-class playbook is to understand all of the science and the clinical results and derive the target product profile to create a best-in-class product. And just to repeat, that target product profile is very clear. No monomer binding. Monomer is very present in the brain. Monomer binding leads to target distraction and reduces efficacy, and monomer is not the problem. No plaque binding. Plaque also creates target distraction. In fact, it may be protective by sequestering oligomers and saving neurons from neuronal death. Plaque binding also contributes to area E, or vascular edema, which is a serious side effect. The key to the target product profile is highly selective binding of toxic oligomers. That allows all your therapeutic ammunition that gets into the brain to go after the root cause of disease and block the ability of those oligomers to cause neuronal death. But it has to be highly selective because oligomers are outnumbered greatly by monomer and plaque. In addition, because we know there are different strains of oligomers, personalized medicine is critical. No single monoclonal antibody will work for all patients. There will be some a priori non-responders whose toxic oligomer doesn't have the epitope target that monoclonal antibody goes after. Well, Promis, based on all of this information and this understanding of the target product profile for best-in-class, well on the way to creating and validating a best-in-class portfolio. We have 5 monoclonal antibodies listed here that we have shown already bind oligomers, don't bind monomer, don't bind plaque, and in vitro or laboratory tests block the processes of disease, neurotoxicity, and propagation. Going forward, and I'll give you a little bit more detail about these things, we're going to show in vivo benefit, blocking neurotoxicity and cognitive deficit in mouse models, do a prevalence study in a cohort, confirm a CSF or spinal fluid and blood-based assay, and show functional differentiation from first-in-class therapies. First, the in vivo model. This is a very well-validated object recognition model. The way it works is this. It's very clever. A mouse is shown one object, and then 7 days later showing that object and a second object. If the mouse's memory is working normally, it'll remember the first object and spend most of its time on the second object. If the mouse's memory has been impaired, it'll divide its time equally. If you look at the chart below, the way that metric works is a score of 0 means the mouse's memory was impaired. It divides its time equally. And that's exactly what you see with the second bar from the left, which is the toxic oligomer bar. The other thing that's very important about this model is it uses toxic oligomers to create the cognitive deficit, which we know is what happens in human disease. If you compare that second bar from the left to the third and fourth bars, what it shows you is that our lead program, PMN 310, completely eliminated the neurotoxicity and the cognitive deficit in this toxic oligomer model. The mouse remembered the first object when they had our drug, despite this presence of oligomers, and spent most of its time on the second object. We plan to repeat this study for the other 4 antibodies. That work is ongoing. We expect our best-in-class therapy portfolio to have significant progress and many key catalysts in the next few months and through the year. First, further in vivo efficacy and information on our lead program, PMN 310, in vivo validation of our other 4 monoclonals, That cohort study could provide extremely interesting and relevant information to prove that precision medicine is critical in Alzheimer's disease. We're looking at the prevalence of different strains of toxic oligomers in over 100 subjects with Alzheimer's disease by studying their spinal fluid and their blood. In addition, we're on track to developing companion diagnostics that we can use to select patients for clinical trials, and a blood-based screening assay that might even identify patients with disease ongoing before they have symptoms. And lastly, we won't spend much of the time of it on this short summary, but we're starting this process with tau and TDP-43 that hopefully will lead to the same sort of a disease-modifying, best-in-class portfolio of products that selectively target the toxic oligomer forms of those two proteins that also cause Alzheimer's and other dementias. Looking at our lead program, PMN310, we're on track towards completing a proof of concept phase 2 study by early 2021. And here are the steps. Manufacturing scale-up and GLP or animal toxicology in 2018, leading to an IND, enabling first clinical studies by the end of 2018. Then a phase 1 safety study in 2019 and a phase 2 proof of concept study complete by early 2021. Now, there's a second benefit to a best-in-class strategy, which is not only do predecessor programs provide you with a lot of the scientific information about the ideal target product profile, but predecessor programs have already gone down the path of development and have provided information that helps you understand the expectations of risk and success going through many of those steps. Let me show you what I mean. Many programs targeting beta amyloid as a monoclonal antibody have successfully gone through GLP toxicology, manufacturing, and phase 1 safety. So that would suggest that our programs have high odds of success. No reason why ours should have different odds than programs that succeeded before us. So we're very much focused on our phase 2 proof of concept. We plan to do a larger study than Biogen's aducanumab and use a companion diagnostic to make sure that everyone in our study has the correct oligomer strain with the epitope that our product PMN310 binds to. Now, what expectations would that leave us? First of all, because of our binding profile, no area E due to no plaque binding, and we'll use an IgG4 isotype. Higher efficacy because we'll be able to dose higher without the dose-limiting toxicity of area E, and higher efficacy due to having a companion diagnostic that screens out a priori non-responders whose oligomer doesn't have the epitope we're targeting. So in summary, we're developing a best-in-class disease-modifying portfolio and are well on our way. Numerous prior clinical programs have contributed to the understanding of what it takes to be best in class, and that is to selectively bind the toxic oligomer, not monomer, not plaque, and to be able to personalize with companion diagnostics. And we have multiple catalysts, data readouts, and value creation coming over the coming months. Over a longer term, our lead program is on track for an IND in late 2018 and phase 2 data coming out in 2021. Just after the likely approval of what we believe will be first-in-class therapy, aducanumab, for the reasons I've mentioned, our scientific advantages, our best-in-class playbook-derived scientific advantages, we believe give us a good chance of coming up with better clinical data right after aducanumab is approved. No area E, efficacy due to the ability to dose higher, and improved efficacy due to better patient selection, leaving out a priori non-responders. Our mission is to make a big difference in Alzheimer's disease, which I know affects just about everyone I know personally, and we believe we're very well along the way to creating a best-in-class portfolio that does exactly that. Thank you for your time and your interest. More information is available on our website. The address is shown here.